guys, welcome to our channel. This is Science Lectures. Thanks for being with us, and you please consider subscribing, you comment in the comment section and like our video please. So we are still continuing with our same biology and we are looking at transport currently. So transport, we already had a video about diffusion, simple diffusion, but in this video we'll talk about some other forms of transport like osmosis. So let's just start off with osmosis. So what is osmosis by definition? So osmosis, when we talk of osmosis, we need to talk about the water molecules. It is movement of water molecules, or you can call them solvent molecules. So it's movement of water molecules. So we are dealing with water molecules. Water molecules from a region of their high concentration, to a region of their low concentration, So we are having water molecules moving from a region where they are high in their concentration to a region where they are low in their concentration. So movement of molecules from a region of their high concentration to a region of their low concentration. But these molecules are water molecules via a semi-permeable membrane. So we can say via a semi-permeable membrane or through a semi-permeable membrane. So first of all, these are water molecules moving from where they are high in concentration to where they are low in concentration, but across that semi-permeable membrane. So that is very important for us to note when we are defining what osmosis is all about. You can also define in terms of the solute concentration, that is movement of water molecules from a region of low solute concentration to a region of high solute concentration. So molecules are moving from a region of low solute concentration to a region of high solute concentration. So in terms of solutes, let's say we use salt, and we have a can. Let's say this is our can, separated by using the semi-permeable membrane. Let's say this is our semi-permeable membrane. A membrane which is semi-permeable, let's say that membrane is semi-permeable. Then I have solution A and solution B. Then if solution A, let's say solution A, and this is solution B, they are the same level. If solution A is 70% salt concentration, has 70% salt, and solution B has 30% salt. And we've defined osmosis as moving from a low solute concentration to a high solute concentration. That means the water molecules will move from region B to region A, because region B is less, region B has limited solutes as compared to region A. So from low solute concentration to high solute concentration, water molecules from a region of their high concentration to a region of their low concentration. So if we have 30% salt, let's assume we have 70% water. And if we have 70% salt, let's assume we have 30% water. So this side we have a high percentage of water molecules, so they move from B to A, and the opposite applies to that. So this is the concept of osmosis, to break it in basic terms. So that this is what happens between region A and region B. But again, we have some other terms to look at before we go further. If a solution has, let's bring in these terms. We have what we call hypertonic hypotonic, and isotonic. So hypertonic, a solution which is hypertonic has high solute concentration, increased solute concentration. Hypotonic has reduced low solute concentration. Isotonic, iso means equal. Iso means the same. So these solutions which are isotonic will have the same. So isotonic, iso means the same. So such solutions will have same concentration. So isotonic is the same concentration. Hypertonic, increased solute concentration. Hypotonic, reduced solute concentration. So if we look at solutions, we'll give these ones, we make it our case study, solution A and solution B. So which one is hypertonic, which one is isotonic? We say hyper means high, okay, hypertonic, hyper, high. So we have increased solute concentration in the solution which is hypertonic. So our hypertonic solution will be solution A because it has 70% solute. 70% solute. So solution A will be hypertonic. Then solution B will be hypotonic. Why is it hypotonic? Because it has a low solute concentration. In other words, it has high water concentration. And water molecules will move from a region of their high concentration, that is hypotonic solution, to the hypertonic solution. How is this one important? If we are to apply it in application in our cell biology. So if we have cells, let's say we are having a red blood cell. Let's say we are having our body cell, which is a red blood cell, and we place it in solution A. Let's say this is our solution A. This is our red blood cell. This is red blood cell, and this is solution A. Solution A. So let's assume this solution has a low concentration. Let's assume this solution anyway is distilled water. Distilled water is this solution. Then the red blood cell also has its cytoplasm inside there. So the cytoplasm of the red blood cell, let's imagine that it is having a high solute concentration. High solute concentration as compared to the distilled water. What will happen? That means the red blood cell will take up water by osmosis. Because we say osmosis is movement of water molecules from a region of their high concentration, or from a low solute concentration to a high solute concentration, or to a region where the water is in a low concentration. So the red blood cell will take up water. So distilled water in this case is now our hypotonic solution. Yeah. Distilled water will be our hypotonic solution. Inside the cell's cytoplasm is where we have the hypertonic solution. So our red blood cell will take up water by osmosis, and what will happen when it keeps on taking up water? It will burst. So this red blood cell will burst because it possesses what we call the cell membrane, and the cell membrane is prone to bursting. So it will burst. Or this bursting. Then we're saying what will happen to the red blood cell. This is cytolysis. So cytolysis. So the cell will burst. That is what will happen to cytolysis. So this is a case study for the red blood cell. Red blood cell placed under the distilled water. It will take up water by osmosis because distilled water has a high water concentration, low solute concentration. Cell will take up water by osmosis, and it will finally burst, what we refer to as cytolysis. The opposite, what will happen if let's assume the red blood cell is placed in another solution now. Let's say this is my red blood cell, and I place it in a solution which is hypertonic, and the red blood cell cytoplasm is hypotonic with a low solute concentration. The cell will, now the opposite will happen, the cell will lose the water by osmosis. And this is, and when it loses water by osmosis is what we call shrinking. It will shrink and shrivel and shrink, something here. Shrink and shrivel because it has lost water. So what happens here is just increase in size, then finally you tear open, you burst because you're taking up excess water and you have a cell membrane which is not resistant to that tug of pressure created inside there. But the opposite will happen here is that this red blood cell is now releasing water to the hypertonic solution. When it releases water, it finally it shrinks. It loses water and it shrinks. So this is what happened. But if this red blood cell again was placed in an isotonic solution, let's say red blood cell cytoplasm, outside solution, they have the equal concentrations. So there will be a net inflow and outflow of water. So the cell will remain literally, will not burst and will not shrink or shrivel. So this is what happens in osmosis as applied to the red blood cell. So the red blood cell, an animal cell, but let's assume, in another case study, let's assume we are having a plant cell. Let's see what will happen for a plant cell. Because a plant cell possesses the cell wall. So let's have a case study where we have cells with a cell wall. So let's imagine we are having our plant cell placed in different solutions of different concentration. Let's say this is my plant cell. This could be the plant cell, then placed in a solution. Let's first consider a solution with a low solute concentration. So let's say this solution is hypotonic, a hypotonic solution as compared to the cytoplasm of the plant cell. So the plant cell cytoplasm will be hypertonic to the outside, to the fluid outside. So what will happen again is that we very well know that water will enter into this cell because water molecules are moving from a region of their high concentration to a region of their low concentration through the semi-permeable membrane. So when they enter, the cell will take up water, but it has the plant cell possesses a cell wall. So a cell wall can resist change in pressure, it can resist that tug of pressure. So what will happen is that the cell will take up water, but it will not burst, and it will become turgid. So our cell wall will become turgid. Why is this relevant? This is relevant. We can have an application in plants, herbaceous plants. So herbaceous plants are capable of having support of their stems because this factor is put into play. Cells take up water by osmosis, become turgid. They become really turgid. So when they become turgid, they are able to support the stem of the herbaceous plant. So this one is very important. The opposite will happen when the water moves out of the cell. So let's look at a study where we have the plant cell again, our plant cell again. But in this case, let's say the plant cell is placed in a solution and is placed in the hypertonic solution. Hypertonic with a high solute concentration as compared to the cytoplasm of this one is hypotonic. So the cell will lose water by osmosis to the outside, the surrounding solution of fluid. So when it loses water, it will lose water and it will become, if it's a plant cell, it will become plasmolysed. So this is what we call plasmolysis. It loses water by osmosis to the exterior, so it will become plasmolysed. And this is what will happen to our plant cell. It will lose water and it will plasmolyze But for the animal cell, we said it will just shrink and shrink. So this one becomes plasmolized. But if a plant cell is placed in a solution which is isotonic, so equal, same concentrations inside cytoplasm and fluid inside, fluid outside. So the plant cell will instead become flaccid. It will become flaccid. So that is what osmosis is all about in application. So different concentrations of fluid will affect the cells differently. So movement of water molecules from a region of their high concentration to a region of their low concentration via semi-permeable membrane is very important, or from low solute concentration to high solute concentration. Our solutions can be hypertonic, they can be isotonic. But more in application, what again we need to know, if we are to apply this in the human cell again, let's imagine we are having our animal cells. If we are having animal cells in the body, and we inject your body with distilled water, what is most likely to happen to your cells? There are animal cells within your body. Let's say you're having some cells within the body, but we inject in distilled water. This is distilled water, which is at a low solute concentration. Your cells can take up this water, and they eventually burst. Take up the water, and they will burst. So what happens in application? If we are to give an infusion, let's say we are giving 0.9% normal saline solution, normal saline, or what is referred to as isotonic saline or physiological saline. If they are to give this infusion, it should have the same concentration as the cytoplasm, the cell cytoplasm. So in other words, the infusion they are putting into your body should be isotonic to the cell cytoplasm. So that is how this can be applied medically. Giving you an infusion of normal saline, or let's say they are giving you IV Ringer's lactate, at least the concentration should be isotonic. If the concentration is not isotonic, you might end up losing the cells in your body. So that is it for osmosis.